And let me, while Sorin getting himself ready, let me introduce Sorin, our speaker. Sorin is professor of computer science, and he leads the Intelligent Systems and Bioinformatics Lab at Wayne State. He holds a joint appointment in the Department of Obstetric and Gynecology, and is associate dean. He serves as an associate dean in the College of Engineering for Innovation and Entrepreneurship. Dr. Draghici is a senior member of IEEE and an editor of IEEE ACM Transactions on Computational Biology and Bioinformatics, Protocols in Bioinformatics, Discoveries Journal, Journal of Biomedicine and Biotechnology, and International Journal of Functional Informatics and Personalized Medicine. His publications include 2 books, Data Analysis Tools for DNA Microarray and Statistics, and Data Analysis for Microarray using R. He published over 150 peer-reviewed journals and conference publications, which gathered over 12,000 citations to date. Sorin also a CEO and founder of a company called Advitya that again works on pathway analysis and genomics and proteomics research. With that, Sorin, you have the mic. Thank you.
Thank you so much. Can you hear me?
Yep, I can hear you. Yeah.
And you see my slides as well, right?
Yes, yes.
Great. Thank you. So I want to tell you a little bit about COVID-19. I know you've heard a lot, too much about it in the past year, but just to set the scene here. So essentially, this SARS-CoV-19 is a so-called the beta coronavirus. It's an RNA virus, and it has some close relatives that we have had to deal with. One is the SARS-CoV, and the other one is the MERS. So those are very similar viruses, very related, very closely related with CoV-2 that we as a species had to deal with a couple of years ago, the MERS. However, the previous 2 occurrences were kind of mild compared to the last one. So the bats appear to be the natural reservoirs. There are some discussions as to maybe it came from a different source, but currently the most accepted hypothesis is that it came from bats. And they were able to kind of switch the host and jump from bats to human. After a lot of hesitation at the beginning, it was clearly shown that it is transmitted by person to person via airborne transmission. So 2 people in the same room are going to be able to transmit one to the other one. And it causes a severe respiratory syndrome. I'm going to go into this a little bit later. Essentially, there are also some bacterial complications. So it's not an easy disease to deal with. As I mentioned, it came from animals. It's a positive single-stranded RNA virus. It's zoonotic, so it came from an animal, possible bats. And so far, there have been over 50 million cases and counting, unfortunately. Also, those 52 million cases brought about at least 1.3 million deaths. And unfortunately, we are still counting there as well. As we all know, the entry route is via the respiratory tract. And then it attacks pretty much all the organs that are throughout the body. So there are effects that can be observed in the lungs, in the gastrointestinal tract, in the brain, in the cardiovascular system, liver, kidneys, in the peripheral circulatory system, as well as in the eyes. As I said, this is not a disease that you want to have if you can do anything to avoid it. You see here some MRI images. So you see some of the effects that it's is doing in the lungs. So you have these opacities. This is it looks like people develop this, have these pieces of glass in the lungs. So essentially it destroys the lung tissue. Essentially it's a very complicated process, but in principle the response to a virus in general happens in the mucosa. So those are the tissues that cover the inside of our nose and the lining of the lungs. So there is a complicated process that I'm not going to go through details, but essentially a virus or a bacteria triggers the release of some cytokine. Then macrophages are involved, neutrophils, and then T cells and B cells. So the immunity essentially has two parts. One is the innate immune system, which is a general response to any foreign organism that comes into the body. And the other one is a more specialized immune system that provides, builds and delivers antibodies specific to that particular organism. So the innate response system is general, is there, it's available all the time, but it's weaker. The specialized one needs a few days for the body to mount that response to provide, to build specific antibodies. But essentially, the epithelium, this lining of the tissues, it's essential in this immunity. So I'm going to take you back in time to March 2020. So it's about a year and 3 months ago. That was the time when the very first wave of COVID-19 hit the United States. The situation was very, very bad. There are about 1,500 people dying every day. The doctors in the emergency rooms, they didn't know how to treat these patients at all. There were phenomena that they had not seen before, including this respiratory failure, including coagulation, and there was nothing that they could do. The only thing was to provide support, to provide liquids, to put people on the ventilators, but in terms of drugs, there was nothing that was available. So the situation was extremely dire. At that time, the first data sets have started to become available, coming from tissue taken from patients. We were able to find some data, and we set up with the following objectives. The first one, we wanted to understand what was the immune response triggered by this virus. The second one, try to understand how this immune response is related to the phenomena that we see in these patients, in particular inflammation, organ failure, and coagulation. And identify the putative mechanisms involved in this particular disease. And if at all possible, identify one or more existing drugs that could be used to treat severe cases of COVID-19. So this is kind of, my background is in engineering, as Farshad mentioned. I'm in a computer science department. So the question is, how on earth a computer scientist can try to do this, because this seems to be something completely in the domain of medicine. And that is the point, I think, of today's talk, because nowadays research in medicine and life sciences is done with lab technologies that generate huge amounts of data. And most of the time, the doctors and the life scientists do not have a background that expose them a lot to such types of data. So they really need us, computer scientists, and you have seen nowadays with the applications of artificial intelligence in so many domains. Well, this is no different. So computer science and artificial intelligence methods are very much called upon to deliver some results in situations in which there is a huge amount of data and other methods fail. So we used a discovery data set, which included triplicates of primary human lung epithelium. This was taken from some patients who died of COVID-19. And it was compared with tissue taken from another person who was healthy, didn't have COVID-19, but they were having the same age. So we're talking about people who are quite old. So essentially, that particular tissue was infected with SARS-CoV-2 and compared with the mock infection with the tissue that was not infected. And there was also a cell line, this A549, which is essentially a cancer cell line, which was also infected with SARS-CoV and compared with the mock infection. Also, the idea was to try to understand what is the difference between this virus and other viruses. So we also looked at data that came from the same A549 cell infected with RSV, which is a rhinosyncytial virus. It's a different respiratory virus. And also IAV, that's the flu virus. Okay, so we have 3 different viruses, one of them being SARS-CoV-2 in this particular cell. So this comparison is going to tell us what is specific to SARS versus the other viruses. And also we have SARS versus mock in real lung epithelium. Also, we got, as I mentioned, lung biopsies actually. So this is whole lung, it's not only lung epithelium, whole lung tissue from patients that died of COVID-19 versus patients that died of other causes and with the lungs being healthy. These are the criteria that we use. We looked at fold changes that were more than 0.58 and p-values more significant than 5%. Those are kind of common thresholds. Let me show you, to give you an idea, I'm going to jump into the software that we developed and used, and I'm going to show what we did directly in the software. I think you still see my slide. I have to switch. Okay. Do you see something that says iPathway Guide at the top left?
Yes.
Perfect. Okay. So this essentially is a dashboard. To the left here, we have some directories in which we organize the data. And I'm going to show you one particular data set. This is the data set that we analyzed. And again, this software took many years to be developed and implemented, and includes a lot of research that we've done over the years. But essentially, what we're looking here is the summary of this particular data set with the top findings in every category. And this is the input. These are the differentially expressed genes. And you can see this is something that I'm doing this analysis for you in live in real time. So this is not prepared in advance, and it's not slides. So this is essentially using the software as it is. So you can mouse over and see what the differentially expressed genes are. And you see there are quite of them. Here, the tabs at the top show the various types of analysis, and they are organized from left to right, from simple to more complex. So you have genes, and then you have biological processes, molecular functions, cellular components, and so on. So let's go to genes. When you go to genes, what you see is that, first of all, there are quite a few differentially expressed genes. This graph shows log fold change. So this is the difference in the expression level of that particular gene. Each dot represents a gene. And the vertical axis is the minus log of the p-value. The p-value tells you how much you trust that particular change. And the horizontal axis gives you the magnitude of the change. Positive fold changes towards the right and negative fold changes towards the left. So you put the thresholds, you put two vertical lines that, and you look outside those lines for thresholds that are larger than this particular value in absolute value. So it was approximately 0.6. And here, 5%, this is a log scale. So you look for minus log p-values that are larger than this. So what's significant is here to the above the horizontal line and outside the vertical lines. So the ones you see colored essentially. The red are upregulated genes, the blue are downregulated genes. And here, of course, you start by looking at the ones that are most downregulated and most upregulated. And you click here, the software is going to show you the gene. It's going to show you what the gene does, the biological processes it's involved in, molecular functions, cellular components, pathways, and drugs that are targeting these specific genes, microRNAs, and SNPs. And if we look at this, we look at the RAS-related glycolysis inhibitor and calcium signal regulator. It doesn't tell us much. And this is the problem why the classical approach fails, because you really need to look at all the genes that are changing. You cannot look at them one at a time. So here, for instance, I'm going to look at the most upregulated genes. So again, follicular dendritic cell secreted protein. No particular pathways, no drugs known to target this particular gene, and so on. So just by looking at the genes one by one, we are not learning much. And that's a problem. That's number one. Number two, there are a very large number of differentially expressed genes. So you cannot just click on the gene one at a time and try to guess what the heck is happening. So this is where the computer science and a more sophisticated analysis is needed. You need to somehow take all these values and try to understand what is happening at the whole system level. So here, let's look for biological processes that are impacted. These biological processes are one category of this GO or gene ontology. The gene ontology is an ontology which is two things. It's a hierarchy of terms that tells you how these terms are related to each other. And you see here a small part of it. This is a biological process, and this response to stimulus is a type of biological process, and immune response is a type of response to stimulus. And this immune response is also a type of immune system process, which is also biological processes. So this is how you read this thing. Now, this analysis that is being done here looks at the number of differentially expressed genes that are associated with the biological process, with a given biological process, let's say immune response here, and compares that number with the proportion of differentially expressed genes associated to this with the proportion of differentially expressed genes found in the entire data set. So here, for instance, there are 1,305 genes associated to this process, and 231 of them are differentially expressed. They are changing in this disease. So we are looking now at the COVID-19 whole lung versus healthy, okay? And the particular assay that was used here measured microRNA values. So this particular proportion, 231 out of 1,300, it's about, I don't know, maybe 15% or so, between 15 and 20%. This is much higher than the proportional differentially expressed genes in the entire data set. And that can be calculated, a probability for that can be calculated, and it's 10 to the minus 24. And that tells you that this particular biological process is very significant. This is how this type of analysis is done. It's called enrichment analysis or over-enrichment analysis, or ORA.
And the problem is that you do this for many, many genes at the same time. So let's look, we are looking now at the top 10. Let's look at the top 100. And you see that they are still giving you very, very significant p-values. Normally, you look for p-values to be lower than 5%. So because there are so many genes that we test at the same time, we need to do something which is called a correction for multiple comparison. There are various types of corrections. The most stringent, the most conservative is called Bonferroni. And even after we do the Bonferroni correction, you see here, there are more than 100 biological processes that are available and that are reported to be significant in this particular disease. So that's a big problem. And this is where kind of the state of the art was when we started our work in this area. So here you have the life scientist could try to understand this disease or any disease for that matter. By the way, everything that I'm showing you is completely general, can be applied for any disease. I'm just showing you what happens in COVID-19. So any life scientist trying to understand the disease has two choices. Essentially, one of them is to look through these biological processes, and there are too many to look in detail and read all the literature for each one of them. So they can look through them and pick the ones that appear to be more interesting to them. But unfortunately, if they do that, this is not good research. Why? Because you will never be able to discover something new. You can only see what you know. You are going to only look to confirm your expectations and your belief. You are not going to be able to discover something new. So that's a problem. The other alternative is, okay, I'm going to look at the very top ones, right? Even if there are 200 or so that are differentially expressed, I'm going to look only at the first 3, 5, 10, whatever, because those are going to be the most important. That's how the thinking went. So if we do that, we look at them, we see immune response, immune system process, defense response, innate immune response. I mentioned briefly about the innate immune response versus the specialized immune response that happen later, right? So if you see the biological processes that are reported to be significant here, they all make a lot of sense, right? So this is clearly a virus or a bacteria that is mounting an attack, and the immune system is responding. The organism is responding with these biological processes that are well known. The problem, so they are all true. They all make sense, but response to other organisms and so on. Absolutely all of them make a lot of sense. But the problem is that they don't provide enough information. These are very general. So if you look in the hierarchy here, as I mentioned, the more general are on top, biological processes, and then they become more and more specialized. And all these things that we are looking here, they are kind of at the top of the hierarchy. So this is the most specific one. So this is the innate immune response, which is a type of immune response, which is a type of immune system process, and so on. But that's the most specialized. And yeah, it's informative a little bit, and it tells us that the innate immune response is active. But we knew that already, because this is the immune response that is. This is the process and the immune response that is involved every time we get infected with a virus or a bacteria. So this type of analysis, it's correct. It's finding things that are true, but it doesn't help us really understand what's happening in this particular disease. So what we did here, we said, okay, let's look at biological processes as they are related to each other by this number of differentially expressed genes. So here we can zoom in into this, and we can see what the problem is. So here you have the first 12 Go terms that have in common not fewer than 79 genes. So that is a lot. Essentially, we are calling various processes as being differentially expressed based on the same genes over and over. So that's a huge degree of redundancy. So we have a different proprietary method of doing this type of analysis. And that is called high specificity pruning. And it's going to give us different types of results. First of all, now we don't see hundreds, we only see maybe 15 or so. These are all the biological processes that are significant. And now we see super specific things. If you see here, now we are going way down into this gene ontology term. So now we are about 8 levels down, and we start to find things that are really informative. So here we are probably 15 levels down. And these are things that start to become specific to this COVID-19. So type 1 interferon signaling pathway, negative regulation of viral genome replication. And then we see some interesting things. You see here, neutrophil degranulation, neutrophil chemotaxis. So say, okay, this seems interesting. Let's look for neutrophils. So these are the two biological processes, but let's look for these neutrophils in the more general analysis. And now we see there are several processes that are related to neutrophils. I'm going to switch back to the slide and tell you a little bit about that. So, let me show you something else before I go there. Let's look at the pathways. The pathways are essentially graphs in which you have nodes that represent genes or proteins that are coded for by genes, and edges that represent signals that go from one gene to another. So this is, again, where the classical approach when we started doing research in this area was to look at the number of differentially expressed genes that fall on the pathway that's just by chance, the same way it was done with the biological processes, and calculate that p-value. So that is a mistake from at least two important points of view. First of all, when you look at the just the number of differentially expressed genes, you ignore the position and the role of the genes on the pathway, as well as the direction and the type of the signals. And the pathway is there to tell you how these things are happening. So to describe the phenomena that are taking place in this organism in a certain very, very strictly controlled way. So you are ignoring essentially all the information that the pathway is supposed to be giving you. So that's one reason for which the classical enrichment is very limited. The other reason is that that p-value is calculated with a statistical model called hypergeometric or Fisher's exact test. And the main assumption of that is that the variables that you are measuring, the genes, in other words, are independent. And here you have the pathway that tells you how these genes depend on each other. This arrow means that the first gene activates the second one, which activates the second one, the third one, and so on. And then, so you have somebody did a lot of research for years to put together this pathway and to show you how these genes depend on each other. And then you kind of turn around and say, oh, let's consider these are independent. So that was very limited and incorrect. But going back to what happens in this COVID-19. So this is a cytokine receptor interaction. The cytokines are these molecules that are produced when there is an infection, and essentially they tell the immune system that there is an infection going on and where that infection is going on. So cytokines and chemokines. And if you see here, there are various types of these cytokines. So these are chemokines, these are class 1 helical cytokines, TNF family, and so on. Red means that these things are upregulated. So you see here that there are very many cytokines that are upregulated, that are being produced. You see here that there are many receptors that are upregulated. And overall, all these types of cytokines promote inflammation. They tell the immune system, hey, you have some problem here. You have a virus or a bacteria, and you start to kick into high gear. And this is what happens. So essentially, you have a situation in which all these signals tell the body that it should increase the inflammation. Inflammation is supposed to bring this immune response, and in general is good if it's not too much. You need inflammation in order to start this immune response. But the problem is that if it's too much, it starts to be a problem. So here is the part that is supposed to control this inflammation and prevent it from getting out of control, getting too bad, too high. And unfortunately, all this part shows that the genes are downregulated. These blue colors mean that they are downregulated. You see them here too. So, so essentially, this is all these genes tell you that you have a lot of inflammation, and all the genes that are supposed to control this inflammation from becoming too much, they are downregulated. So this is essentially the picture of a freight train going down a slope with no brakes. These are the genes that are supposed to be acting as the brake. So this tells you that in this particular condition, you have a condition which is called hyperinflammation or cytokine storm. The body produces so many cytokines that, and you have inflammation everywhere, that it starts to attack the internal organs, and those organs are going to start failing. And essentially the patient doesn't die because of the virus itself, it dies because of the immune system of its own body, right? So that's the first important conclusion that we find here. So the first one, as I said, in this case, COVID-19 brings this, we're talking severe cases. These are people who died from this disease, right? So there is a lot of hyperinflammation, and essentially have a cytokine storm. The second one, the second piece of information that we were able to learn from the data was that neutrophils play a crucial role. And this is a slide that essentially tells you what happens with the cytokine storm. So this is the pathogen, in this case is SARS-CoV-2. Those are the chemokines, cytokines that are in the inflammatory signals telling the body to produce dendritic cells, macrophages, monocytes, and so on. And unfortunately, this process is so powerful that it produces this cytokine storm that leads to multi-organ failure and hyperinflammation syndrome and death eventually. So as I said, so it is kind of ironic, but this is the reality, that it's not the virus that kills you, it's your own immune response system that is going to kill you. Let's talk about neutrophils. Neutrophils are a very, very interesting type of cells. They are part of the immune system. So what they do, they die when there is an immune response. They die for the purpose of kind of releasing the DNA and forming these nets. Essentially, all the DNA that is contained in the neutrophils essentially is going to tangle with other pieces of DNA and is going to form a net that is supposed to contain the foreign organism. So it's a different type of cellular death. It's kind of they are committing suicide essentially for the good of the organism of the other cells, right? This is an image. Actually, this is a microscope. Electron microscopy image, and you see these nets that are formed with the DNA material from neutrophils that died, right? So this is a neutrophil that died. So there's another programmed cell death, which is called apoptosis. This is different. It's called netosis. And as I mentioned, the goal here, the apoptosis is a phenomenon that happens when the control mechanism within the cells discover that the cell doesn't work properly, and then the cell has to die again for the good of the community. Otherwise, you have cancer and other things, right? But in this case, these neutrophils are committing suicide, you know, like kamikaze, right? So they sacrifice themselves in order to fight the enemy. So essentially you have these very complicated effects of this. So now you have to have this platelet activation. So this has to do with the coagulation. So essentially that hypercoagulation that is seen in COVID-19 patients is due not necessarily to the classical coagulation process that happens when you cut your body somewhere and then the blood starts gushing up and eventually is going to coagulate. And the purpose there is to close the wound and prevent additional blood loss and start the healing underneath. This is a completely different process and is due to, in part, to these nets formed by the neutrophils that are dying. Okay, so this is essentially another explanation of another phenomenon that is seen in the COVID-19, the fact that you have this coagulation problem. So I was talking to an emergency physician who was treating these COVID-19 patients in the early days, and they were just shocked. How he said, we just put this line in their arm, and you know, less than a minute later, it's already coagulated. The phenomenon is so rapid. Okay, I don't want to go into too many details here, but essentially you have this thrombosis and coagulation that is another major cause of complications, let's say. But the ultimate cause here is that hyperinflammation. So inflammation, and now you have platelet activation, which takes you to those coagulation phenomena. Let's go back to the software and let's see what else we can see. So, so you have seen how the data itself, if you do the right analysis, the data itself starts to tell you what is happening in this disease, okay? Let's look at the second pathway in terms of significance. Here, because the mathematical model that we developed in our research, instead of looking, as I mentioned, at the number of differentially expressed genes and comparing with what you get by chance, it's also looking at where the perturbation happens at the level of every gene in the pathway. So now you can start looking at coherent cascades. Those are putative mechanisms that tells you the phenomena that are taking place. And here, these things identified this particular mechanism. And you see here, those are all chemokines outside the cells, which are types of cytokines. And they are all upregulated. The receptor is upregulated as well. And then the next gene downstream is called JAK3, is upregulated, and STAT1 is also upregulated. And you see these other edges here that are highlighted. So essentially, this type of analysis is able to identify the mechanism on the pathway that is actually involved in this disease. And essentially, I'm not going to go too much into details, but if you look at this gene here, we are trying to figure out what was the connection with this. This is kind of, it's all one together. The edges are in a sequence, but this is kind of separated. So the question is why? And if we look here, this is a network that includes only the genes that are differentially expressed and kind of is independent of the pathway. And now we can look for that particular gene, for instance, this FGR, and we see that there is actually an edge between that and the cytokines. And if we click on the edge to see what kind of evidence we have for that, in this case, there is not much evidence, but I think I'm looking at the wrong gene. FGR, PIK, limb, okay. So CCL1. Okay, so essentially, I cannot find it right now. I don't remember which one it is, but essentially there is, those two are connected. And the issue here is that the pathway does not reflect that knowledge yet, but the knowledge exists in the literature. So essentially this forms a single mechanism. Now, the interesting thing is that this chemokine JAK-STAT mechanism is actually there also in other diseases, such as influenza. This is the usual flu. And if you see here, you have the interferon alpha, which is upregulated and was found to be important in COVID-19. And you have the JAK-STAT IRF9 pathway. So this seems to be a mechanism that is actually very important in this COVID-19. Let's look at upstream regulators. Upstream regulators are genes that are changing, and their particular change triggers, controls other genes downstreams. So in this case, IRF9 seems to be extremely highly significant. You see here, I'm going to correct with Bonferroni, is 10 to the minus 10 after the most stringent correction known to mankind, humankind. So IRF9 is important, STAT2 is important. So again, the JAK-STAT mechanism, and interferon gamma, interferon beta. So those are the really important genes. So essentially, this here shows that this particular gene controls, I don't know, maybe 15 genes. I don't want to count them right now, immediately downstream. But let me cut to the chase. So the whole purpose here is, yes, to understand the mechanism, but also to find a drug. So we have the capability to look at existing drugs and see which of those existing drugs could revert the changes that are induced by the disease. So I'm going to look for drugs that are predicted as being absent or insufficient. So if I add this drug, I'm going to kind of change this, the changes induced by the disease and revert them, bring the body to closer to the normal. And if you look at them, you start to see things that are very interesting. Tofacitinib, this is a drug that I can go to a drug bank and show you. So it's a JAK inhibitor, this Janus Heinous inhibitor, and is used in to control the inflammation. And diclofenac is an NSAID, so it's a non-steroidal anti-inflammatory agent, so similar to ibuprofen, but using a different mechanism. Methylprednisolone is a steroid, again, used to reduce the inflammation. I'm going to just show you only the approved drugs. And then gold sodium thiomalate, again, is something used for anti-inflammatory action in the treatment of RA. So these are drugs that all control the inflammation, and rheumatoid arthritis has a lot of inflammation. So you start to look at drugs, and there are not that many, and they all make sense. So essentially, you have a hyperinflammation, you have the cytokine storm. So this is what you need to worry about. Now, we also went and we looked at the other types of tissue and other viruses, as I mentioned before. And this is a meta-analysis. And if we focus on this and we can look at the, whoops, let me go to the drugs. I was showing you genes. So I'm going to look for drugs that can be used to revert the phenotype. I'm going to focus on the lung epithelium and look what the #1 drug is. And this is significant in every one of the 5 experiments that we have here. And we verify that in other experiments as well. So methylprednisolone is the drug that is extremely highly significant. It's able to revert the changes in real patient tissue, in lung epithelium, as well as in other in other infections. But this is the ones that we care about. So there was a problem. So we found the drug. So we found mechanism, upstream regulator, and drugs, but there was a problem. The problem is, I'll show you in a second, those are the mechanisms that we find. I don't want to go into too many details, but the problem is that these are the other types of cell that we used. So we use Vero-E6 cells infected with SARS. We use lung organoids. We use intestine organoids. So to see whether these phenomena are also happening in other types of tissues in the body. So lung, intestine, and others. So the bottom line, as I mentioned, these are the validation results, and you see the drugs are exactly the same, which was absolutely astonishing, right? Because these are independent, different types of tissue obtained from different patients in different labs and processed in different labs around the country. So to find exactly the same drugs was absolutely astonishing and gave us a lot of confidence. So we chose this methylprednisolone. And the small problem that I was mentioning is the World Health Organization. At that time, this is the official guidance from the World Health Organization as of May 2020. And it says corticoid therapy is not recommended in SARS-CoV-2. Okay, so here we had a situation in which the whole world, the smartest brains, the smartest doctors in the whole world were saying that steroids are not to be used. And our work, our research showed that actually they could save lives. So what do? We do a clinical trial. So this was a clinical trial that was undertaken together at Henry Ford Health Systems. It was led by Dr. Ramesh. It involved 213 patients diagnosed with COVID-19. And about 38% received the conventional therapy and 62% received this methylprednisolone. And we looked at the patients getting worse, which means that once they are admitted into the hospital, do they need to go into the intensive care unit? Or if they are in the intensive care unit, do they need to be put on mechanical ventilation? Or if they are on a ventilator, do they actually die? Okay, so this is a composite endpoint that is looking at these three things. And what we found was, again, amazing. This drug was able to reduce the mortality from 29.6% to 16.6%. That's 44% reduction in mortality. And in these situations, in order to keep the research as objective as possible, people look at 30-day all-cause mortality. So essentially, even if the patient gets well, is sent home, they still follow up to make sure that they didn't die later. And they include all-cause mortality. So even if they got hit by a bus, you still include it in this kind of study, right? And you see there's a huge difference that people with the standard treatment, which was just supportive, died at a much higher rate. So this is the number of people who are alive at the beginning. And then every time somebody dies, the line goes down. So the red is the methylprednisolone group. The other one is the control group. So shortly after we did this work, we had a press release, which was largely ignored because I think people didn't really want to believe that this is happening, that the methylprednisolone is effective. But shortly after that, there was a study from the UK that looked at dexamethasone. And that was done in, I think, 30,000 people. So we had 200 people. So maybe that's why they didn't take it, they didn't change the current practice. Some people did, and some doctors did, and they changed a lot of, they saved a lot of lives, but not everybody did. But once this study from the UK came out, it showed that another steroid, dexamethasone, was effective. Now the question is, what do you use? Do you use methylprednisolone or dexamethasone? People are using dexamethasone, but the dexamethasone actually requires more, this NNT is the number needed to treat. So you need to treat 25 patients in order to save one death. Okay, that's the standard way of looking at the efficacy of a drug. Whereas for methylprednisolone, you only need to treat 8, so which means that it's much, much more effective. So anyway, but the bottom line is that we were able to find a drug that will reduce the mortality by 44%. And now, well, not now, but since September 2020, so a few months after that, now the recommendation from the World Health Organization is that, yes, we recommend systemic corticosteroids rather than no corticosteroids. So they changed their recommendation based on this. We published this in peer-reviewed papers. So these are, the first one is the one about the mechanism, the ones that I talked about today that identify methylprednisolone. This is the result of the clinical study itself. This is just as a clinical study. This is presents our results in thrombosis and vascular injury, so coagulation and so on. And this is effect on kidneys. So those are 4 papers that we published in this area. We'll be happy if anybody is interested to look at this data or do additional analysis. Just send an email to this address, and we'll be happy to give you access to the platform and the data. So that's it. Thank you so much for your attention. I'll be happy to take some questions if there are any.
All right, Sorin, thank you. Let's see if any question from our audience this morning. Everybody's quiet. I'm assuming, Sorin, everybody got their vaccination, so they don't need to be worried about any of this.
Right. Yeah, that's good. That's good. That's what we want. We don't want to get people on the last breath and then save their lives. We want them not to get sick in the first place.
All right. So this work that you've done, let me ask you, I know your earlier effort in this genomic world was in pathway analysis. Can you tell us a little bit about how you started in this field and become interested in more genomics, proteomics research?
Sure.
Bioinformatics in general?
Yeah, so as far as I mentioned, I'm a faculty in computer science. My background is entirely in computer science. I've bachelor, master's, and PhD all in computer science. And my PhD was actually in artificial intelligence. This was a long time ago in the mid-90s. And in neural networks in particular, on which I did my PhDs, you are dealing with situations in which you have very many variables and not very many samples, which is very different from what the classical statistics deals with. In a classical statistic, it's exactly the opposite. So you have very many samples, you have hundreds or thousands of samples and a few variables, like 10 variables and thousands of samples. So the classical statistic doesn't offer the instruments and the tools to analyze this data. And nowadays, as everybody knows, there is a huge explosion of data in all areas, and you have this term of big data, and essentially this happens in life sciences. I was attracted to this area because life sciences are an area in which what you do has a huge impact. As I showed you here, this is a situation in which our work identified a drug that actually saved lives. It reduced mortality by 44%. So this is huge. So I felt, for me personally, I was looking for this meaning for the work and the research that I'm doing. So that's one area, one reason for which I went towards life sciences. The other reason was very opportunistic. So I realized that in life sciences, they have these huge data sets coming from lab techniques that produce many, many measurements. And the people working in doing research in that area don't have the right background. So it's very difficult for them. So there is a huge opportunity for a computer scientist, somebody who chooses to study computer science and this advanced, artificial intelligence and advanced data analysis and big data. There's a huge opportunity for us to have a contribution and help people who do research in life sciences. And at the same time, we have the this very rewarding part that what we do is very meaningful, right? I have the feeling that, you know, if I were to work in a different area, what I do would have an impact, maybe even if it doesn't matter what you do, but it's going to affect just, you know, maybe you have a better better engine, or, you know, faster compiler and so on, but really does not impact directly the people's lives and health. And I think that's what drew me to this particular area of activity, of research.
Appreciate it, thank you. So let's see if there's any other question coming from audience. All right. No questions. Seemed like you lost the audience, or they were so impressed with the work. Again, you mentioned your earlier work started in neural network. And I also mentioned in my introduction that we are looking into introducing a degree program, a master's degree program in AI and machine learning. And some of the work that you are doing, of course, taking advantage of these technologies, correct? So the question is that, how do you see such program will evolve in the college that cuts across many disciplines, correct? Is it informatics, health informatics, medical informatics? Is it to do with the work that we are doing in autonomous driving and technologies, in data science application that we have in the college?
Yeah. So I think the beautiful thing about this, that at the end, regardless of the domain or the specific application, everything comes down to some numbers. So so you have some big data sets, essentially. You have many variables and some measurements for them, and the goal can be slightly different from one application to another, but in all situations you end up analyzing data. So and I think this is why such a degree would be very, very important and very useful to people, because once you learn the tools and the techniques that are available in this area, so you have the weapons that you can use to attack any problem whatsoever. So it gives you a set of skills that is amazingly versatile. So you can work and you can go and work for an automobile company like Ford or Tesla or something like that, or you can work in healthcare, or you can work in any area, because at the end you have the same. You have a huge matrix with numbers, and the question there is, it always, you need to build a model to learn from some data set and try to predict what happens with new data sets, or you need to control something. But the nice thing is that they are all done in the same way. So once you learn certain basic techniques and the way you think and the way you attack a problem like this, then the problem domain is less important. So you can use the same knowledge in many areas. So you are going to be a really very desirable set of skills. So I think such a degree is going to be, number one, very important, and number two, very useful to a huge number of people.
Yeah, you're going to become very, very competitive in the marketplace with this set of skills.
I think so. I think we had many students that went through our programs in computer science and in electrical and industrial engineering, who studied different activities, but now they're specifically focusing on AI machine learning, and now they're data scientists and doing work for, again, many industries that we have. How many students are you working with currently? PhD students and masters and undergraduates and so on?
So last year I had like 3 students graduating. I have another one graduating in the summer, we hope. And we currently have 5 PhD students in my group, and I think we have 2 or even 3 maybe newly admitted students who will start in the fall. So unfortunately, they are coming from abroad, and there are some issues related to the COVID-19 and the visa and the ability of the embassy and consular offices and so on. But essentially, so we have 5 current PhD students, and we're going to get some more. We are always looking for interesting students. So if anybody is interested to learn more about what we are doing, or maybe to join the graduate program in this area, please get in touch.
Sorin, tell me about, again, you mentioned you're a computer scientist and you get into this bioinformatics. One of your first PhD students who graduated in computer science under your supervision, where is he now?
So we have, we graduated quite a few PhD students. One of them is a tenure professor at Stanford University. Another one is a research faculty at Stanford as well. We have another PhD student who graduated from my lab, who's a faculty at the University of Nevada. We have, so essentially, we have somebody who's in at Amazon. We have people at Netflix. We have people at Microsoft, and so on. So they are going in the to the top companies and top universities in the country and beyond.
I appreciate it. I think that shows the quality of the work that is being done by you and your colleagues, and really putting our name on the map with having outstanding graduates working for various higher education institutions or global companies such as Microsoft, Amazon, and so on. All right. Any other questions that you may have, people? I know.
Rashad, this is Azad. Can you hear me?
Yes, Azad,
yes.
I can hear you.
Yeah, hi, Sorin. Nice talk. So I was thinking about, because I come from electrical engineering background. So currently I'm in the mechanical engineering department. So for me, the size of the data is an interesting question. So what's the size of data that you actually need to make a legible decision and basically a prediction of what kind of drug is the most useful? So, and also, do you rely on just the medical records, or you actually also use the patient's information? So for example, if you have an app collecting data from patients, so I know, so for example, CDC for the vaccine, they are collecting some data on weekly basis. So do you actually have used those kind of data? So it's 2 questions. So what's the size of data to make a reliable prediction? And then if you have used any data from the patients?
Yeah, so all the data that I showed today came from, well, not all of them was real data. But the one that I actually showed you was all from the patients. We had the other data sets that I mentioned, but all the results that I showed you in the software, those are all coming from the patients. The other coming from cell lines and tissues, organoids. But so in that sense, the data that we use most of the time comes from the patients. How big the data? So how much data, how many measurements do you need? So it really depends on what you are trying to accomplish. If you, in machine learning and artificial intelligence, you have a couple of different scenarios, and there are 2 big branches. One of them is supervised machine learning, and the other one is unsupervised machine learning. In the supervised machine learning, you some labels, and the goal is to build a model that allows you to predict the labels for new samples. There, you have to have a correct ratio between the number of dimensions in the model and the number of data points, right? So the data that I showed you today for COVID-19, those were mRNA, so gene expression measurements. So there are about 30,000 variables.
across different number of patients. We commonly, so that's the crucial point, the 30,000 variables, right? So if you are talking about unsupervised learning, then actually the more data you have, the better, because unsupervised learning is going to build a model based on the redundancies between the classes. So let's say if you have only 3 samples and each of them is different, you can never do anything with unsupervised machine learning. So you need to have, like, tens or hundreds from those 3 classes, so that the machine learning has the opportunity to distinguish what's in common between those 3 groups. So it's not an easy question to answer that, but in general, the whole area deals with, let's say, tens of thousands of variables. That's the number of variables that we typically use. And again, the number of, so you have 2 dimensions. One is the number of variables. The other one is the number of samples that you have. So so we use usually tens or hundreds of samples and tens of thousands of variables. So that's why we are doing kind of machine learning rather than statistics, as I mentioned at the beginning. Did I answer your question?
Yes, thank you. And then one other question, as you said, so as you have the more, like, increase the data, so it improves the accuracy. So like, did you actually continue, like, did you add data, like, to see what is improved over time? So for example, so you get some more data from different geographical locations. So does it actually bring down the prediction, or like your suggestion, to a certain number? So in other words, so can you actually say this is the lowest that we can go, or this is the highest that we can go? And then even if you have the whole data in the world, you cannot go beyond this. So is that, like, is there something like there is a limit to the prediction that you can provide based on the size of the data, or it can be actually improved as you increase the size of the data?
Yeah, so it really depends what the final goal is. So in this particular work that I showed you here, the goal was to find the drug. That's what we set up with. So again, if you roll back in your head what was happening about a year and a couple of months ago, when there was no treatment available for this, and doctors didn't know what to do with these patients. They were dying like flies left and right. So our goal here was to find the drug, and we found that drug. It was the methylprednisolone. Can you see my screen?
Yes.
Yeah, I can see your screen.
So it was the methylprednisolone. So, but before you go into any kind of human clinical trials, you really have to have a lot of validation. And these are the data sets that we validated on. As I mentioned, there are different types of tissue coming from different labs and so on. So once we get the same kind of results on this, my mouse doesn't work anymore. On these additional data sets, there was nothing else to be done at this point. So it doesn't matter how much, as you said, you can have all the data in the world. I mean, we identified the drug, and once it was verified in 3 other data types, in 3 other types of tissue coming from 3 different labs and so on, there is nothing more you can do. So we were ready to go and propose it to the doctors. And once you verified in a clinical trial, it doesn't matter. It doesn't matter how much data you have. Then you are going to get the same results. You are going to point to the same drug. It's not that the drug is going to. You are going to find a better drug if you have more patients.
Sorin, are you there? Hey, Sorin?
I think we might have lost him.
Yeah, it seemed like we lost him. So it was not me. I thought it was something bad. Let's give him a second. I think he's gone. Yeah. Let's see, he's gonna dial back in. So while we're waiting for Sorin to see if he's gonna come back in or not, you can certainly, if you have any more questions or interested, working with him on some future projects, he had provided us with his contact information. You can also try to contact me or Kirsten here, so we can make sure that your message gets to him so that he can respond to your questions or future inquiries about his program and the work that he's doing in the Department of Computer Science. Next week, we will continue with this series of talks. We have Dr. Kaishang Wang from the Department of Electrical and Computer Engineering, who's going to be talking about sustainable, reliable, resilient, and affordable electric energy development and delivery, a hybrid and diversified approach. So I invite you all to join us again next week. Again, the same 9 o'clock talk by Dr. Kaishang Wang from the Department of Electrical and Computer Engineering. With that, I'm not seeing Sorin. Do you have a last minute question?
Sorry, I'm back. My computer just decided to, crash.
Of course, Sorin. So Sorin is back. Any last question for Sorin before his computer start rebooting and installing software updates?
No, I'm back up.
All right. Any questions? All right, Sorin. I want to thank you again for your excellent presentation. And again, that's a good way for us to showcase our faculty to our friends and partners and alumni globally. And I look forward to seeing you guys next week at the same time for the talk by Dr. Kaishang Wang. Thanks again. Have a wonderful Friday. Bye-bye.
Thank you.
Thank you, Sorin.
Don, did you have a question?
Yeah, my wife works in the College of Pharmacy at Wayne State, and she was working on MRSA and more recently COVID, and I think she would find this really, really interesting.
Yeah, I responded to the chat, Don. So feel free to put her in touch with me. If she sends me an email, we can set up a time, and we'll be happy to share with her what we did and answer any questions she may have.
Okay.
I also have another webinar that we did with one of my co-authors on one of these papers, and that would be probably better as well, because it's more on the life science. I try to keep this kind of understandable, so I didn't want to go into the life sciences too much. But sure, I'll be very happy to talk to her.
My personal experience with prednisone, I had a lung issue several years ago, and the stuff is just phenomenal.
Yeah. And it was kind of very interesting to see. It was a very, very interesting experience in general, because here was a situation in which the data clearly showed that this drug is going to work, right? And we even had a clinical trial results showing that it does work. And yet, the initial response was very lukewarm from the clinicians. They just wouldn't believe it. And this was because, again, the World Health Organization recommended at the beginning not to use it. So But it was very clear to us. I mean, once you look at the data, it was very clear what was happening. You have a cytokine storm. You know, the asteroid would be ideal. The interesting thing that we can also show, I didn't have the time to go into it, but not all steroids are the same. So there are the steroids that are not expected to work so well because they didn't revert so many of those changes induced by the disease. So that's another interesting, it's a whole different discussion that for this audience, it didn't make sense to go in that direction, but that's really, really important. So.
You think about things probably a little over a year ago, the ER doctors had no idea how to deal with this.
Yeah, exactly. And that's what we wanted to do. We tried to help with that. And it was very interesting because we had responses that range from, these are ER doctors, right? So emergency physicians, right? And we had people who said, oh, of course, and jump on it right away and say, oh, that makes a lot of sense. And we had people who said, oh, don't you dare to say that, and you know, you cannot really have any recommendation before it's peer review. And I said, but we have clinical data and we have results in patients. Oh, no, you have to be peer reviewed and you are irresponsible. We had a press release about this, and you are irresponsible to have to put out this press release and so on. People really got upset. And that tells you that, you know, there's also the human factor, right?
Yeah.
So.
Well, thank you.
Sure. Thank you so much. And again, I'll be very happy to talk to your wife.
Okay.
So, all right. Thanks a lot.
Yeah.
Bye.
Bye-bye.
Have a nice day.
Thank you. Bye-bye.